Hi, my name is Ava Miciuda, and I go to Troy High. And this summer I worked in Dr. Zong's lab, and my project was testing mouse Sphinx2 and Spag6 interaction by gene princeps luciferase complementation. Okay, so we studied the Spag6 or mammalian sperm-associated antigen 6 gene, and that is found in cilia and flagella and encodes an axon central apparatus protein. So there are actually two very similar genes, SPAG6 and SPAG6L, that are found on different chromosomes and only a few amino acids different, and they have completely different functions. So when SPAG6 was knocked out, there were no disorders noticed, and SPAG6L is known to regulate ciliary beating, but it also has many other functions, including regulation of celiogenesis and spermatogenesis, hearing, and immunological synapse. So here are the two SPAG6 proteins, and here you can see the amino acid sequence, and all the highlighted letters are the ones that differ between SPAG6 and SPAG6L, which are not very many. And over here you can see the protein structures, and again, they're very similar. There's only a couple differences. Okay, so why do these two SPAG6 proteins have different functions? Both SPAG6 proteins have armadillo repeats, which are known to mediate protein-to-protein interactions. And we performed a yeast hybrid screen and used SPAG6L as a bait. And we identified several binding partners, which were SPINK2, SNAP-in, SPAG16S, and SPAG16L. So the original goal of the study was to compare the SPAG6 and SPAG6L binding partners with the protein complementation assay. And here are all the functions of the proteins. So the protein complementation assay to monitor protein-to-protein interaction. So how it works is there's two proteins. One has an N terminus of luciferase and the other has the C terminus of luciferase. So separately, they express like little to no luciferase, but if they interact, then they together express the full length of luciferase. So there'll be a lot of luciferase activity. So that's how you can test to see if proteins interact with each other. So here's the project outline. So what we did, started with PCR and then did TA cloning, plasmid isolation and digestion, sequencing, subcloning, transfection, and then Western blot and luciferase assay. Okay, so first PCR. So we designed primers for our target cDNA, and then we amplified the sequence using PCR. Then we ran it through a gel to get our specific gene of interest and to separate any other non-specific bindings. Then we did TA cloning, which is ligating our gene of interest into a plasmid. So we did this and put it into bacteria and grew them on a kanamycin plate because the plasmids are resistant to kanamycin. So you can see here on this, we also put IPTG on the plate. So that turns the colonies without our insert blue, and then all the other ones will be white. So we know which ones we can use to then isolate and digest. And we did that using the enzyme EcoR1. And then here's the digestion. So you can see the TA vector and then the insert. Then we prepared them to send to sequencing to make sure there are no mutations. And we did subcloning. So subcloning is a transfer of DNA inserts from one vector to another. So we used the DNA that we just sequenced and had no mutations, and we transferred it from the TA vector to the target vectors that express either the C terminus or N terminus of luciferase. And then we digested them using the enzymes HIN3 and AGE1. So here again is the digestion results, the vector, and then the insert. Then we did transfection. Transfection is introducing purified nucleic acids into eukaryotic cells. So we put our gene into HEK293 cells. Then we did a Western blot to detect the specific proteins and cell lysate. So here we use the SPINK2 antibody, and in non-transfected cells, you can see there's not really anything there, which is good, and same with SPAG6 but in the cells that we did transfect with SPINK2, it's present, so that means our results can be trusted. Then here's the luciferase assay we did to see if proteins activate or suppress the gene. So here you can see the non-transfected cells had very low luciferase activity, and that's the control. And then SPINK2 and SPAG6 by themselves also had little to no activity. But when in the cells that had them both transfected, there was very high luciferase activity, meaning that they interact a lot. So future plans, using this, we can compare the difference between the difference of protein interactions between STAG6 and STAG6L by the luciferase assay. This will help us figure out why STAG6 and STAG6L have different functions. So here are acknowledgements. Thank you to everybody in the lab and everybody else.